by Professor K. Ramachandran Reddy, and second lecture by the Professor Neeraj Singh. The technical session will chair by Dr. Dr. Sasi Khan Singh. Dr. Sasi Khan Singh, the M. Pharma PhD, currently working as a professor at the Faculty of Pharmaceutical Sciences, Mahayogi Gorakhnath University, Gorakhpur. Dr. Singh published more than 9 research papers in various reputed international journals like Elsevier, Tyler, and Francis and Bentham Sciences. In his credit, 2 books were published and have 2 patents. His research expertise in phytochemistry, in silico drug designing and discovery, and drug use in the management of Alzheimer's. Dr. Singh served as a reviewer for the various renowned journals. I would like to invite Dr. Sasi on the stage. Please welcome Dr. Sasi, sir. This technical session, our co-chair is Dr. P.K. Chaudhary. Dr. P.K. Chaudhary served as a professor at Varanasi College of Pharmacy. He is gradually acquired highest degree M. Pharma in Quality Assurance Technique from Bharati Vidyapit University, Pune College of Pharmacy and Research Center, Pune, and hold the PhD degree from Berhampur University, Odisha. He has 8 years of technical experience. He joined Sri Vittal Education and Research Institute, College of Pharmacy, Padarpur, as a principal. After that, he joined Royal College of Pharmacy, Education and Research, Malegaon, Maharashtra. Further, he joined as a professor at Varanasi College of Pharmacy. He had also around 6 years of industrial experience in different institutes like United Engineers Cuttack, Akhil Pharma Private Limited, Hyderabad, AP Qualiquare Pharmaceuticals Limited, Hyderabad. He has participated in a different workshop cum training program on instrumental method of chemical and drug analysis. His research finding has also been presented at a reputed national conference and his research work published in peer-reviewed journal. I welcome to Dr. Chaudhary. Please come on the stage. I request Dr. Pradeep Kumar Chaudhary to facilitate our chair with memento and certificate. Now, lecture series will be started. Is hand over to Achal.
Now today's our first speaker of ninth technical session is Professor K. Ramachand Reddy sir. Professor K. Ramachand Reddy sir, working as professor and head department of Rashastra Institute of Medical Science in Banaras Hindu University. Dr. Reddy gradually acquired highest degree of BMS, MD in Ayurveda, PhD in Ayurveda, HPA education clad in Japan, D literature. All these degrees are pertaining to Ayurveda, Rashastra and Bhaisatya Kalpana from various universities of India and abroad. Dr. Reddy wrote 24 great Ayurveda books in Sanskrit and English. Dr. Reddy have tried to establish Ayurveda in scientific manner at international level. Dr. Reddy have published more than 19 research papers in international peer-reviewed journals with high impact factor. Professor K. Ramachandra Reddy sir is well known for valuable contribution in the field of Ayurveda pertaining to Ayurvedic pharmaceuticals, Rashastra and Bhaisajya Kalpana. Further, from 2018 to March 2020, Dr. Reddy also worked as member of Ayurvedic Pharmacopoeia Committee, Unani Pharmacopoeia Committee, Siddha Pharmacopoeia Committee, Homeopathic Pharmacopoeia Committee, United Pharmacopoeia Committee. He is also appointed 2 times as head Department of Rashastra as Bhai Sajya Kalpana, 2 times of Superintendent of Ayurvedic Pharmacy, Faculty of Ayurveda, Institute of Medical Science, PHU Varanasi. At present, Dr. Reddy is continuing as Professor, Department of Rashastra, Bhaisajya Kalpana, Faculty of Ayurveda, Institute of Medical Science, Banaras Hindu University, Varanasi, and presently, Dr. Reddy is collaborations of allopathic doctors developed various Ayurveda formulations which are antivirus in nature and effective against. The entire project is led by you and your team. With this, I request you, sir, to please come on this stage and enhance our knowledge on the topic designing and development of Ayurvedic dosage forms of management of SARS-CoV-19.
Okay. Namaskar. Respected Chairman of the session, Dr. Shashikant Singh, respected Co-Chairman, Dr. P.K. Chaudhary, coming from Banaras Pharmacy College, respected Dean, Faculty of Allied Sciences, Dr. Sunil Singh, in the absentia, Honorable Vice Chancellor and respected Registrar, Dr. Pradeep Raoji, and the whole team of this organizing committee, which is struggling for since last 2 months to organize in a better manner. And I really appreciate for this total best organization, particularly Dr. Sunilji. I am thankful to the organizers for giving this opportunity. Before starting my session, I would like to brief regarding the Ayurveda medicines and how Ayurveda medicines also we are doing scientific research to enumerate the details pertaining to the components of the Ayurveda medicine as well as their therapeutic action and also how the mechanism of the medicine taking place in the body system to affect the disease condition and also to maintain the health in the healthy person. As you know that Ayurveda contains three genomics, Vata, Pitta and Kapha. Based on three genomics, the constitution of the body of the person will be evaluated and the disease condition will be understood by various techniques like Nadi Pariksha, etc., diagnostic parameters. And based on all those diagnostic parameters, the disease will be treated by medicines which are produced by various raw materials which are procured natural from natural resources. In Ayurveda, we procure medicines from three basic natural resources. Abundant quantity of materials are the plant materials. Second quantity of the round about 98% are the plant materials, and 1% may be the animal products, and less than 1% are the mineral products. So basically, plant, animal, and mineral origin materials we use for making the medicines in Ayurveda. These materials are taken and processed by different pharmaceutical techniques. For example, the minerals are concerned, we have a very separate pharmaceutical technology called Basma procedure. We process the material into Basma, which is acceptable form to the body system, and such Basmas in very lower dosages will be administered to the patient. Thereafter, the therapeutic effect will be produced. Now, the situation is that various diseases has been described in Ayurveda and their treatment also has been described. But sometimes we face new diseases and the treatment method has to be invented for the new diseases in Ayurveda system. Similarly, SARS-CoV-2, the COVID-19 is the one of the new outcome in the disease processing, particularly infectious disease, which is came into the picture very suddenly and affected a large population in the world. Now, the Ayurveda mentions that whenever disease is not known to the Ayurveda physician, based on the parameters of three genomics, that is Vata, Pitta, and Kapha, their domination of provocation and involvement in the body system, the disease will be identified and that disease will be cured as per the pharmacotherapeutics mentioned in Ayurveda. Like modern system, how the drug components are explained, in Ayurveda also, the pharmacology is very well established. We call Panchamahabhuta, Rasaguna Virya Vipak, Prabhav and Karma. These theories are simultaneously coordinate with the provoked Vata, Pitta and Kapha genomics. So based on this, in Banaras Hindu University Institute of Medical Sciences, we developed certain medicines for the treatment of COVID in that period, and we have administered the medicines. And this study was done with the collaboration of allopathic general medicine unit and also Ayurveda. Myself, I was leading this project. The project was approved by Human Animal Ethical Committee, later on Animal Ethical Committee, and further we also registered in Clinical Trial Registration of India, that is CTR. All the protocols has been followed. Thereafter, we have studied the total medicine. So in this project, three major components are there. The one part, we have worked a clinical study of the medicine, of Ayurveda medicine for prevention of the COVID-19 like infectious disease. Second phase is that curative part, that is curative part for treating the medicines with the medicines the infected patients. We have isolated wards in the Institute of Medical Sciences and we got the abundant number of the patients also. And third part is that post COVID treatment. Here, since the all the clinical study parts of treatment of COVID-19 and post-COVID treatment and the basic study includes a very elaborative part. Since in this conference I was allocated the time period, based on the time I will be focusing on the three main components, that is the preventive aspect of the SARS-CoV-2 and antiviral study of the tablet and also the genome study. These three phases will be discussed now. In a coming future, the clinical study part of the patient's care also will be discussed in any coming future occasion. I think, can I ask the students how many Ayurveda students are present over here? Can you raise your hand? How many biotechnology and other allied science students are there? Very good, very good. Thank you. So it is very nice to see all the science students who are having interest in Ayurveda. So in the prevention, as you know that in Ayurveda, there are a huge number of dosage forms has been explained. Most of the dosage forms, because of time factor, they become very outdated and they are out of the practice also. Most of the medicines presently we are using tablet, churnas and basma, etc. But in this situation, even in COVID period, I have selected one particular dosage form that we call it Dhup Kalpana. Dhup Kalpana indicates the fumigation of the medicinal plant drugs. After burning the medicinal plant drugs, the whatever the fumes are coming, they will sterilize the nanoparticle size of that medicine present in the fume is having less diameter than the RNA virus, so that it penetrates and it destroys the virus. And also the most important channel of body system is that nose, mouth, and eyes. These are the pathways where the virus is going to get entry into the body system. So these pathways were going to making the entry of the virus. Then we have seen that in Ayurveda, this is the one of the best medicament where by exposing the fumes, we can destroy the viral entry into the body system so that we can prevent the medicament, prevent the disease. And once the whichever the virus is entered all into the upper respiratory tract, again, because of inhalation, this dupa will destroy in the upper respiratory tract also. So this study was done with the clinical study, and actually the study also done by different phases. One is analytical part of this DUPA. Second one is safety study over the animals, rodents. Here I am not going to present safety study over the rodents, albino rats and mice, because it will be very elaborative. And third one we have done over the Drosophila model also. Over the Drosophila model, we manufactured a chamber and we introduced the dhupa, and we have given the Drosophila to inhale that dhupa, and we have seen how they sustain and their oxidative stress and toxicity, etc. parameters were screened. So this Dhupa in those days of COVID period to manufacture in our institute became very difficult. So that is why we have requested one of the well-known Ayurveda industry called ML Pharmaceutical India Limited, and they have manufactured for me, as I have given the design of the formula, and the Dhupa was manufactured by Delhi Ayurveda industry, and that was taken to for this study. So here I will be presenting over the Drosophila model, how the Dhupa was studied, and in clinical study, Institute of Medical Sciences, how Dhupa is used. further the other part of the study will be discussed. So this is the basic idea. The stick of the medicinal plants has been made by Emil Pharmaceutical India Limited, and it will be burned and it will be inhaled through nose and mouth so that the viral entry will be prevented. So, to evaluate the efficacy of this air Vaidya herbal dhupa, the nomenclature also has been given by me, and the various parameters we have fixed, and this is the formula of this dhupa we have prepared. This contains all the plant materials. These plant materials in various proportion has been given, and these plant material, most of the plant materials are already proved as antiviral drugs. Some of the plant materials also proved as anti COVID, and most of the plant materials are already proved as antibacterial also. So these plant materials in particular ratio, as per my experience, has been collected. They may contain like turmeric, tulsi, etc., well-known drugs, and loban like the fumigation material, gummy material also used. I think you might have seen in Muslim culture, they will give some dupa that contains loban. So that is basically originated from Ayurveda system of medicine. So we have done parallel single arm blind randomized controlled trial. Uh, 150 cases subjects we have taken from the Institute of Medical Sciences patients, and in intervention group, and whereas in control group we have taken the only 100 subjects. And this is the total flow chart, how we have given the medicines. In OPD, we have selected the subjects, and thereafter the individuals are screened, and in intervention group, we have given the DUPA. And this DUPA is given in 10 by 10 room by giving fixed measurement. The patient will be asked to inhale one complete stick for around about 15 minutes in morning and also in evening. Then like that the study was went, and these are the inclusion criteria. The persons from age 18 years, and how the patients are excluded, all those things are followed as per the protocol of the clinical study. The later on follow-up study also we have done up to 2 months period, and no patient got affected with COVID after its treatment, this treatment in intervention group. The number of people found to have RT-PCR positive in COVID-19 infection, both in intervention and control groups, to evaluate the efficacy of intervention. We have taken RT-PCR as one of the parameter, and these are the other toxicity study. Any medicine, even though it is a herbal medicine, it is the need that we have to say whether it is safe or not. So safety is a first, efficacy is secondary. So that is why we have done safety study on two parts. One is over the albino rats and mice that I am not presenting here. Here I am presenting over the Drosophila model in with the collaboration of biochemistry department of Banaras Hindu University. We have done this Drosophila model. We have done the safety study as well as oxidative stress measurement also. Everywhere we got very good results because most of the tulsi like plant materials, they have the very good volatile property. Even the Drosophila also is ready to inhale, and it is accepting the fumes and does not have any of the adverse effects. All the statistical data, how number of flies we have taken in each group. We have done the fecundity study and different other assays in the biochemistry over the Drosophila model. So after doing the that safety study, when it is found safe, then the clinical trial was conducted. As I told already, 150 subjects we have screened, and all the subjects we have found they are very safe. So with these parameters, we came to know this is very safe. The fumes are very safe because the safety study, as per Drugs and Cosmetic Act, only over the animal is sufficient. But we have done over 3 species. One is albino rats, and also Swiss mice, and also Drosophila model. The reason is that to completely evaluate the safety study, because there should not be any question, because most of the modern doctors raise it that your dupa may have the carbon, and carbon inhalation is not acceptable. So initially, the ethical approval also became very problem. And after explaining about the traditional medicine, after my tedious explanation, they have accepted in the Human Ethical Committee, and however, it is approved and registered in CTRI, CSIR organization also. So in the Drosophila model, that is why we have done all this oxidative stress measurement and laying status. Everywhere the results are very good, and this is the parameters we have studied: fever, cough, cold, anosmia, loss of taste in intervention group as well as control group. You can see that a small bar is the intervention group where there is no symptomology, etc., because of exposure with the dupa. Whereas in control group, we have found all these symptoms. So all the parameters based on the statistical analysis proved that this is very effective in the prevention of the COVID-19. So I mean to say that the Ayurveda medicines, if like as I have taken the help of biochemistry in Banaras Hindu University and general medicine, here you have the allied sciences, biotechnology also. If you have Ayurveda and biotechnology collaborative work, you can explore many of the medicines scientifically. With all the proofs, you can show the world and their evidences for the benefit of the human. So this is the justification of our study. So ancient dosage forms like dhupa, which is even not known to even Ayurvedic doctors also, because nowadays it is outdated. So that we try to bring back, and that too in a situation of COVID-19 like that, where it is more explorative and also a serious condition where patients are going to get death also. So in such a situation, we have taken it as a challenge, and we evaluated all these things and proved its preventive role in Ayurveda by using Ayurveda system of medicine. Further, we have done the clinical study. For clinical study, we have this is the dupa manufactured by the after this study. This is manufactured in large scale all over India, and it came in newspapers of all over India, and various channels also has released its news. and it was sold in a large quantity. I am very much thankful to the ML Pharmaceutical India Limited, who taken my ideas and accepted. The director immediately accepted in such a COVID period itself, and they put investment of lakhs of rupees to develop this formulation in a most scientific manner. We have made a platform we have made a stick of the medicinal drug, and it will stand the patient or the subject person any place in the office or any place. They can keep over the table and they just burn it and inhale it. So it will purify the room. Later on, people also notice that the mosquitoes also not coming and there is no other viral infection also. And later on, some of my friends from dentistry, they asked me that for dentistry, during the dental teeth treatment, we have to expose with a certain fumes. Why can't you develop these fumes for our dentistry? Then we accepted, and we are under the underway of developing its usage for dentistry also. So, like this has been developed with a pure Ayurvedic idea only, and try to bring up the ancient Ayurvedic outdated dosage form, that is Dhupakalpa. Now coming to the curative part. In curative part, we have given this Dhupa again patients who are affected for the COVID-19, and we have developed a tablet for internal administration. Along with tablet, one syrup also we have developed Ayurveda syrup. And in one group of patients that was given, and in another group another tablet which is already in the market of ML Pharmaceutical India Limited, that is Pepetro. This tablet is called Swarnavir, is designed and developed by me, and the tablet was again formulated, manufactured by the ML Pharmaceutical India Limited. So before giving in the clinical study, we have done this tablet, we have evaluated this tablet for the safety study over the animals again with albino rats and mice, both two species of rodents. And again, we have done the study of the antiviral study over the cell line. And the under the antiviral study, we have done three phases of the study. One is the therapeutic study of this Swarnavir tablet, where we have given the we have infected the cell with the COVID virus, SARS-CoV virus, live virus, and we have given the tablet and we have seen the effect. Second study we have done is the virucidal activity, in which we have done the direct virus and tablet components by in the form of solution reacted, and how the tablet components are destroying the virus under the phase of the virucidal activity has been revealed. And third one is cytotoxicity study over the cells, how the Swarnavir tablet has been produced toxicity or not, whether it is safe or not, that also we have revealed. And in this way, the total study was published in Springer Nature, journal, PubMed journal, Springer Nature. And this study I would like to present before you. As you know that SARS-CoV is one of the pandemic and infectious disease condition. And in this study, what we have done is that, first of all, I would like to mention that why Swarnavir tablet nomenclature has been given. Swarnavir, like remdesivir, Vir for virus, and Swarna is that it contains 98% herbal drugs like Guduchi, Tinospora cordifolia, etc. And apart from that, we have incorporated gold basma in this formulation. That is, gold is called Swarna in Sanskrit. That is why we call it Swarnavir tablet. And this Swarnavir tablet has been manufactured by Emil Pharmaceutical India Limited by after my formula submission, and they have given me the prepared tablets. The tablets we have studied for this antiviral study, and all the drugs, and also here Guduchi Tinospora cardifolia. The plant material is abundant quantity. The plant we have not used as such. In Ayurveda, there is a particular method of water soluble component extraction of any plant material that we call it Guduchi Sattva. Guduchi Sattva means the Tinospora cardifolia plant, which is stem, which is having equivalent to the size of thumb. That much stout stems will be taken and they will be cut into small pieces, mixed in 10 times of water, crushed manually, and all the starch and also water soluble component which is dissolved in the water, that components mixture of water will be taken away, decanted, and that water will be evaporated. Whatever the fine soluble, water soluble matter is there, that we call it Guduchi Sattva. This Guduchi Sattva in Ayurveda shops you can get as a Guduchi Sattva. It is abundantly used by Ayurveda physicians for the treatment of fever, like paracetamol. We use Guduchi Sattva for reduction of the temperature and fever. So that is why in COVID-19, fever, cough, these are the anosmia, these are the major symptoms. Based on the symptoms, the formulation has been designed and the Guduchi Sattva has been taken and in abundant quantity. So this is the formula you see. Guduchi Sattva is 140 mg and it contains some of the classical medicines like Mahalakshmi Vilas Ras, Swaskas Chintamani, Jai Mangal Ras, and Yashad Basma. Yashad is zinc. Zinc we use in the form of basma. Zinc, even in modern medicine also, they use for antiviral treatment as well as skin treatment and many respiratory tract infections also. So that is why this formula specifically has been designed. And these particles, how they work, all are proved. Individual drugs, they are proved for antiviral nature and they are published in high impact journals also. And in this study, as I told, in vitro study, we have screened for therapeutic effect, and also this study was done on live COVID virus itself, because in those days live COVID virus was not permitted by all institutions. Only central government has given permission to certain institutions, like one of the institution is at Pune. So with the permission of the central government in Pune, I conducted this study over the live COVID virus. The species of the virus, genus, everything has been mentioned here, and the solution how we have made, at what temperature and what condition we have given, the virucidal activity, therapeutic activity we have studied, and all those things are mentioned here. You can take this slide and you can see. And another thing is that this is published in open access Springer Nature Journal. This is all the matter is available over the mobile itself. You can log in by K. Ramchandra Reddy BHU anti-COVID study. It will display itself by Springer Nature. So all the details are in the public domain. It can be taken and studied at any time, and how the study was given, all the details are given here. And this study found that this Swarnavir tablet has been proved as a very potent therapeutic agent for the SARS-CoV-2, and also it is safe because of cytotoxicity study, and it destroys the virus because of its components, and it is safe for it doesn't spoil the physiological system of the body system. And during the study, all the cytotoxicity details also revealed, and how the samples has been taken, and how many 72 hours we have incubated, all the details are given. It is published also. And these are the 2 hours therapeutic mode, 1 hour therapeutic mode, and 4 hours, and virucidal mode. Everything we have recorded, and all the details are given in the published paper. So, with this, Ayurveda drugs, when studied for the uh, studied for the uh, any infective disease or chronic disease, depending upon the requirement of the public, uh, definitely you will able to find out very best therapeutic agent from the Ayurveda system. So I request all of you, because since you people are from scientific background, so with your collaboration, Ayurveda can develop very efficient medicines. During the phase of clinical study in Banaras Hindu University, we have central government has given one of the center to screen the virus nature, to study the virus genome. We have the genome bank also in Institute of Medical Sciences. So with their help of allopathy doctors, in my clinical study, the sputum of the patients was collected, and their virus we have studied. And during my clinical study period, the virus has been found that it is Omicron genome. So that study also we have published in Q2 journal, which is PubMed, Scopus, and Web of Science also. So this is the drug metabolism journal. It has been published. Ayurveda medicines in alleviating the symptoms of COVID 19 Omicron variant in North India, because in Banaras region, the genome study which we have studied, that is found to be Omicron that period, that also we have published, and Omicron variant was isolated and noticed, and submitted these details to the government also, our clinical study. And with all these information and rest of the clinical study in any other occasion, I will be submitting to you all. And with given time, I would like to conclude all the information of my study here. So the outdated dosage forms like Dupa, etc., has to bring back, and the medicines which are having therapeutic effect in certain diseases like COVID-19 also can be evaluated, and all the opportunity was given to me in Banaras Hindu University. That is because of great founder Pandit Madan Mohan Malviji, who was very great visionary of this educational system uh, present day established in Banaras Hindu University. who was the teacher of Mahatma Gandhi also, a great freedom fighter, the Pandit Madan Mohan Malviya, because of whom we could able to work in Banaras Hindu University, a very big university, which is the first biggest university in the Asia and fourth biggest, largest university in the world. So I was happened to be a faculty member and senior professor of that university. I'm happy and also grateful to the my founder of the institution, great Madan Mohan Malviya. I'm very much thankful to the organizers, particularly Dr. Sunil Singh, Dean of Allied Sciences, and all the team, all his team, and also the chairman and co-chairman, and all the respected members, faculties present over here, and all the students who are hearing this session very patiently. Thank you very much. Namaskar.
Sir, actually, the fumes is liberated from the stick. तो उसका कंपोजिशन क्या है सर? गैस का उसमें लिमिट होता है।
कौन सा? एक बार रिपीट करें।
सर वो फ्यूम जो एलिमिनेट होता है
हाँ हाँ
तो उसका कंपोजिशन क्या है सर?
हाँ, एक्चुअली जो कंपोजिशन जो है we have solidified the fumes, and those fumes we have done X-ray diffraction, scanning electron microscopy, and transmission electron microscopy study in our Banaras Hindu University. That is another study because this study was very much elaborated. So I could not present all the analytical part also. Those fumes contains various inorganic substances, very less carbon. And also, I would like to bring to your knowledge is that unlike other material, wood material, if we burn, it will release many of the ash. It will retain some of the ash. But this medicinal drug, when we burn it, the remaining portion does not contain much ash. The fumes contains various elements. We also noticed that zinc also was present. Many of the components inorganic, because fumes doesn't contain any particle of organic nature. All organic matter will be burned, and inorganic substance which can tolerate the temperature that will spread in the form of fume. And we also try to find that the diameter of the fume and the virus, the diameter, how the fume nano size is able to penetrate the particles of the fume, how they can penetrate into the virus, and how these particles are going to destroy. For example, if I burn here, this area become disinfected. How it happens? In ancient day, one of our great surgeon called Susutacharya, during operation, the operation theater will be sterilized by this type of fumes. And after that, they will start the surgery. So the basic idea is that sterilization, destroying all the microorganisms, including virus, bacteria, or any organism, airborne organism, which is spreaded in the air. So that is why we have studied a long way the components of the fumes and their components, and how these components are going to penetrate into the virus, and what is the nano size of the components, and how they enter into the virus, all the mechanism we are going to publish into another paper. So that is why till its publication, I didn't submitted all those details. Whatever the details submitted here, only published matter which is accepted by the journals like Springer Nature or any Q2 journal or Q1 journal only presented here. So the underway research which is going to be published in any coming future regarding the components and their elaborative study which we have done, that also we will be submitting. At present, let me tell you, it is very interesting question. The thing is that the fumes contains inorganic micro nano-sized elements which will penetrate into the virus, bacteria, and they are responsible for the sterilization of the area where it is released, and the how much area it is going to be spreaded by burning one stick, that also we have studied, that will be submitted. Thank you. Thank you for it.
Sir, is there any sign and symptoms to the immunocompromised patient or any allergic reaction of this?
This inhalation. I have already presented. The intervention group of the clinical study contains 150 patients. Control group contains 100 patients. In intervention group, the signs and symptoms of the COVID like cough, fever, and anosmia has been reduced, and there is no any adverse effects. We have the Pharmacovigilance Center also in Banaras in the university. ADR, adverse drug reaction, if it is there, that we will record in that. That is one part. Apart from that, we have designed a chambers. In that fixed size transparent chamber, we have kept three animals at one time, albino rats, and we have given one stick of fumes, burned, and we have seen their behavioral nature, and later on after study, their organs also studied for the toxicity. There is no adverse effect. And again to reveal, we also studied Swiss mice in same chamber. So two types of rodents are studied. Further, it is also studied Drosophila model, which I have already explained here. In Drosophila model also, there is no adverse effect. Moreover, they have sustained. Their respiratory capacity has been increased. The tolerance also very good. Oxidative stress parameter also good, that I have shown all the statistical parameter. So based on all these safety studies, there is no adverse effects, and based on the clinical study over the subjects, there is no adverse effects found. The reason is that all the medicaments are the plant material like tulasi, loban, etc. These plant materials contains volatile and also other medicaments. When they burn, their inorganic substance in the form of fumes will be released, and that fumes are found to be very safe. Since ages, these fumes by the name of dhupa, as I told, the great surgeon of Ayurveda, Susuta, has used this dhupa for the sterilization of the operation theater. Later also, when newborn child is born in Ayurveda, we expose this dhupa to sterilize that infant baby, to avoid any infection to that baby. So such type of practices are there. They are outdated present day. I try to bring back that dhupa, in that to particularly COVID-19 like a situation we took as a challenge. The same question asked by you is also asked by me, by general medicine professors when we took this study before getting ethical committee approval. Apart from this question, many other questions are there. like carbon, etc. We have explained after doing all these safety studies only we are permitted this study for the subjects human study, and this study was registered in CTRI, that is Clinical Trial Registration of India, and after that only it has been done.
Thank you so much, sir. To enhance our knowledge about outdated doses of the drug regarding this very interesting class COVID-19. Thank you. Thank you very much.
Now I would like to request our Dean Sir, Allied Health Science, to come on this stage and facilitate esteemed speaker with certificate and memento.
I am the
Give a big round of applause.
Thank you for nice organization and also giving me the opportunity. Thank you. Thank you, Chair. Namaste.
Thank you so much, sir. Now our second speaker of third day of national seminar is Dr. Neeraj Sinha, sir. Dr. Neeraj Sinha, sir, you are highly accomplished and respected professional in the field of biomedical research from CBMR, which formerly known as Center of Biomedical Magnetic Resonance. Dr. Sinha have received his Master of Science from Jawaharlal Nehru University and PhD from Indian Institute of Science, Bangalore in 2003. He had completed his postdoctoral fellow in 2003 to 2004 Iowa State University, USA. His total research publication in the per-received international journals is 109. Dr. Sinha have viewed nuclear articles and book chapters such as nuclear magnetic resonance based on metabolomics from cancer research. Solid state NMR spectroscopy, the magic wand to view bone of nanoscopic resolution. Cross polarization applied to the study of liquid crystalline ordering. He attended several invited talks, participation in conferences, departmental workshops, schools, and he done PhD thesis, supervision of many PhD scholar. Dr. Neeraj Sinha sir, research group primarily focus on methodology developments and applications of NMR spectroscopy to understand the intricate biological systems and the different inherent biological processes governing human health. With this, I request you, sir, to please come on the stage and enhance our knowledge on the topic NMR-based metabolomes from biomarker discovery to drug development.
Okay. Thank you very much. Good morning. Good morning. And first of all, thank you very much for the organizer to inviting me to this very beautiful campus and very interesting conference, BioNatureCon. All the organizers have done an excellent job in arranging such a conference. And I come from Lucknow, Center for Biomedical Research, which is in Sanjay Gandhi Postgraduate Institute of Medical Sciences campus. And my research area is metabolomics. So I will try to introduce you what is metabolomics. And of course, NMR is one of the technique, nuclear magnetic resonance, which is a very important technique for doing metabolomics studies. But there are other spectroscopic methods also, which can be applied for metabolomic studies. So metabolomics is a branch of science, omic science. Omic science in the sense that you have a genomics, you might have heard about it, and you have proteomics, transcriptomics. In the end, there is a metabolomics. So this is something, a new technique which has emerged for last 20, 25 years. I will try to introduce you what this technique is, how it works, and how it can provide some very important information about the disease system. And it can be helpful in detecting a biomarker. And very recently, it has also been applied to drug discovery. So drug discovery is a branch of science where you try to find a new drugs for the particular disease. So let's start like how this metabolomics work. So you know that this biological system is a very complex system. Human body is very complex. There are so many things which happens in our body, and all the time you eat food, it get digested, it goes to the nutrition go to the blood, and then finally through different organs you perform functions over the years, you age. So biological system or human system are very complex in this sense. So metabolomics allow us to look at these things in a more holistic point of view. For example, like you have a genomics. Genomics is basically a study of genes. You study different genes in a system, and based on these genes, you figure out what are the genetic sequences responsible for a particular disease. So it's a branch of gene. But in a biological system, what gene does is that it translates the genomic information through transcriptomics to RNA. So RNA is here. So gene encodes the RNA. And in the biological system, finally, this RNA makes the protein. So if you are studying the protein in the entire system, it is called as proteomics. But in the end of all those biological process, there are small molecules like metabolites, which does the biological functions. So if you are looking at those small molecular metabolites in a complete fashion, we call this as a study of metabolomics. Okay, so with genomics, we can get a lot of information. With transcriptomics, we can get a different kind of information. If there are certain disease, then certain transcription factors are unexpressed. And if there are, if you are studying all kinds of proteins in the biological system, and then which does the main function, then it is called as proteomics. But the newer branch of omics sciences is looking at the metabolome. Metabolomes are the end product of the biological system. So if liver is functioning, that means it will be in that process there will be a lot of molecules which will be generated. If the liver function is affected because of certain disease, then definitely those signature will be present in those small molecule or metabolite. So this is one such example. So these small metabolome can be anything like sugar molecule, nucleotide, amino acids, lipids, antibiotic, pigment, and all those things. So what happens that because of this diet and lifestyle, this our metabolome changes, or disease conditions, like if some disease is developed in a human system, then also metabolome will be affected. And if there is a drug or toxic stimuli, you give some drug to the patient, then also because this drug affect the entire process, and the end product is metabolome, so metabolome will also get affected. Or some external stimuli conditions you give, then it also gets affected. So the idea of metabolomic is to measure all the molecule and compare it with a standard system to figure out what are the changes which has happened. And based on those changes, you figure out what are the metabolic pathway which gets affected because of diet and lifestyle, or some disease condition, or drug or toxic stimuli, or environmental stimuli. And based on those analysis, you find out what is happening in the biological system. So this is one of the main idea of doing metabolomics. Metabolomics is not just limited to a certain branch of biology, but even in the Ayurveda, people have started applying metabolomic studies to understand how a particular treatment or drug response or the things are happening for the biological system. So one thing you have understood that we need to measure the small molecule metabolome as a function of this biological process and how it happens into that. So let us see how we can design our study to do a very good metabolomics. So one thing is there, if you are going to study a certain system, whether it's a disease system, drug system, or Ayurvedic system, you need to first understand what you are going to study. Okay, so first part is study design, which comes in the first part. So you have to design your study in such a way that what is your risk group, what is the disease group, what can be your inclusion criteria, exclusion criteria, what can be control system, and all those things. Okay. Suppose I am interested in studying the diabetes patients, how the metabolome happens in the diabetes patient. So I need to include my group as a diabetic patient. Control should be non-diabetic. My ages should be comparable and all those things. So why I'm telling all these things? Because you people are going to be the future scientists. You will graduate in Ayurveda or whatever field you are trying to do. You yourself can design your study depending upon your interest. So this is how it is being done. Now, once you have decided about your study group and inclusion criteria and all those things, you need to decide what kind of biofluid you are going to use. So this metabolome will also be there, present in blood, urine, or any of the biofluid which is being secreted by the human. So your choice of biofluid is very important, like which should show the signature of disease condition or your point of study. So you can use mini valve. Mini valve is one of the fluid which is collected from the human lung, and I'll show some example of mini valve study in my research, which we currently carry out in Lucknow. You can take serum, blood serum. Blood is a very good biofluid which can be collected from patients, and then you can measure the metabolites, or plasma, or urine septum, depending upon the kind of study, exhale, breath condensate, CSF. All those are the very important biofluid which can be used. Well, once you decided about the biofluid, you have to collect those samples from the patient and then process it in such a way that your results gives exact information about your study. Then comes the NMR acquisition. NMR is nuclear magnetic resonance, one of the technique which will allow you to detect small molecules with a very high accuracy, very high range, not only qualitatively but quantitatively also. So I'll show some examples about this also. And then once you have acquired the NMR spectrum, then you need to process your data in such a way that you get information about the molecular information about the molecules present in this patient. And then comes the very important part, which is the chemometric analysis. So you will be collecting hundreds of samples, hundreds of molecules will be identified, and based on those, you are going to compare disease group with a control group. So you need to apply very good statistical tools, and there are very good statistical tools being developed for the metabolomics purpose, and these are called chemometric. And once you get that, you get a lot of information about the metabolite which are responsible and then metabolic pathway. Then you need to have a biomarkers, and this will give you biomarkers which are responsible for the disease condition. And then there are other biological analysis which you can carry out. So I'll give you one particular example where for the metabolomics case. Well, NMR is one such, spectroscopy is one such method, but there are other spectroscopic methods like GCMS, LCMS, and they all have certain advantages and certain disadvantages. So now people are going for more holistic way where they are not stick with one technique, but they use all the three techniques in combination with each other for the metabolomic studies. Okay. So I'm going to show one particular example of metabolomics in a critical care setting. Critical care is a very important section of the tertiary care hospital where patients are there in mechanical ventilator. And I'll try to show one example that how we applied metabolomics to get the biomarker and to tackle the problem of critical care. So in critical care departments, patients comes in a very late stage of their medical treatment, and their lung pathophysiology is severely affected, and because of that, they are put on mechanical ventilator. So ventilator is there, which is a machine, because their lungs cannot absorb the oxygen, so they have to be put on a high pressure oxygen into that. So this is ventilator does that. And ventilator not only does that, it also keeps on monitoring very important vital parameters for the patients, and like how much oxygen has been inscribed and how much oxygen has been given. So based on this oxygen pressure given and how much it has gone, they calculate this P by F ratio, the ratio between partial oxygen in the artery blood and fraction of inspired oxygen. And this ratio is a very good indicator of hypoxemia. And if this ratio is very bad, that means patient is in a very severe condition. If this ratio is good, very large value, that means lungs are performing very good oxygenation. So what happens that based on this P by F ratio, which I am calling at P by F ratio, like how much oxygen is given. And recently, this disease has been defined based on this P by F ratio, which is, we call it as acute respiratory distress syndrome, ARDS. If P by F ratio is 200 to 300, then ARDS is mild. If it is 100 to 200, then it is moderate. And if it is less than 100, then it is very severe. Okay, so based on this P by F ratio and this classification, clinician give them therapies like so that the patient can recover. So ARDS has a very different heterogeneity, like it can come from sepsis, pneumonia, transfusion, and all these other different conditions. So one of the problem in critical care is like when the patient comes, they may be mild, and certain patients recover and go out from ventilator, but certain patients become from mild to moderate and severe very fast, and finally they die. So one of the challenges in critical care is to how to get a better prediction whether the patients from mild are going to be in severe or finally the mortality condition, so that the clinician can change their course of action. Even though the PYF in mild, they may start the severe treatment if they know beforehand. So this is exactly what we did by NMR-based metabolomics, trying to figure out if we can get some markers which can be very predictive in nature. So there are clinical scores like Apache and SOFA, which are very good indicator for clinical mortality and severity. And outcome is and mortality outcome is uncertain and mortality is elusive with this Apache and SOFA score. So what we did was we collected one small fluid from the lungs. So this lungs is under the severe condition, so we can collect one fluid which is called as bronchoalveolar lavage fluid, and then we can take the serum also, and we can do NMR of both. So all these biofluid will give us information about the lungs condition, and with a very high accuracy we can measure them and we can study their changes. So the idea is to collect both mini bulk coming from the lung and the serum, and then do based on this subphenotyping of ARDS by using NMR, if we can get some metabolic endotype, which can be a very good indicator for classification between different stages of ARDS and how we can predict that. So we designed our study. Again, I told you that study design is very important. What would be inclusion criteria, how many samples, and all those things, and how we are going to collect the samples. So, and then based on these different numbers, and finally, our aim was to get the metabolic endotype and some predictor for the mortality based on all those molecules which we measured by this method. So this is how the NMR spectra looks like. You see, first is the serum spectrum. Serum means blood. For ARDS, you can see there are so many different molecules which can be identified from the serum, and this is the control. Control means no ARDS. So you can see that all these molecules are very highly expressed because of ARDS condition. And this is the BALF. BALF is a fluid which is collected from the human lung. So even in the human lung, we can measure a lot of molecules which are very high concentration because of ARDS. And if we compare with the control, then this is very high. So you see, these are the metabolites which are expressed because of ARDS condition. So by comparing and a statistical analysis, we can find out if there is any molecule which is a very good biomarker and how it can be used as that. So statistical analysis comes into the picture for the mini valve, for the control and ARDS. We can get a very good statistical analysis for the separation. And for the serum also, we can get the very good separation. Then for the different stages of mild, moderate, and severe ARDS, we can identify the molecule which are responsible for a very good separation between that. So you see that these are the different molecules which are very good indicator for separation through our statistical analysis. So tyrosine, tryptophan, leucine, threonine, these are amino acids which our lungs is secreting because of that. And similarly for the other kind of biofluid also, we can get that. So for the mini valve, pulmonary and non-pulmonary ARDS conditions can also be separated, and these are the molecule biomarker for the separation. And similarly for the pulmonary serum based, we can get a very good separation. So based on all these analysis, we have identified set of metabolites from in the mini valve as well as on the serum, which can separate the two phenotype of the ARDS condition. So in the mini valve, it is lysine, arginine, isoleucine, and alanine, and all those. So these are the metabolites which is responsible for separation between the two. And these are the metabolites or biomarker discovery which has been achieved in our study. Now comes the whether we can use these metabolites as a predictor for severity, whether the patient is going to die or not, or going to survive. Can the measurement of these molecules be used as an indicator for survival or not survival? So this is something which has a lot of potential because we have found that there are molecules in mini valve like isoleucine, tyrosine, which are very good predictor for mortality between survivor and non-survivor. Okay, and in the serum also there are these molecules which are very good predictor for survival and non-survival. So with this, we could find out that these are the molecules which are predictor for survival or non-survival. What does it mean? It means that if you can measure those things and categorize over them, you can predict whether this concentration is higher than the certain level, it will be patient is going to die. So you have to start more aggressive treatment, even though P by F ratio is very less. So this is how things work. Now with the clinical score like Apache and SOFA, which clinician use that, survivor and non-survivor group can also be done in a very good predictive manner. But if we apply our metabolites with Apache and SOFA, we can say that it's a very good, much better predictor in terms of mortality and things like that. For example, like leucine is gives a very good cutoff between the survivor and non-survivor. So if non-survivor people, if isoleucine is beyond this level, that means the patient is not going to survive if we continue with the conventional treatment. So they have to start with that. So this is how one of the example, like how the metabolomics can be used to measure the biomarker. Okay. So, so in our studies, we have combined certain molecules which are very good predictor for endotype combined with clinical scores. And not only that, we also get information about the biological pathways which are affected in disease condition. For example, like here we see that arginine and proline metabolism is affected in ARDS, or all these metabolic pathway are also, information is also attained because of metabolomic study. And similarly for the minibar sample, we get the different amino acids metabolic pathway, how it is getting affected into that. So metabolomics not only just gives you the biomarker, but it also gives you better understanding of the disease by looking at those pathways which are affected. So in terms of combined our analysis, we found that it is the TCA cycle which gets mainly affected in the ARDS condition, mild, moderate, severe. And these are the different pathways which are affected and how these molecules are linked in terms of the pathway. So this is giving a more holistic approach toward understanding the ARDS and also leading us to clinician to target those pathways if they want to have a better uh, management in the ARDS condition. So one thing is like getting the good biomarker about the disease condition. Second thing is pathway, and then leading it to the understanding like how to tackle a particular disease by looking at those pathways. So metabolomics can be used into that. So this is another such example which I would like to highlight. When the ARDS is also present with some comorbidity conditions like AKI, acute kidney injury. So you see ARDS is basically from the lungs and kidney is from the other part of the body. But sometimes what happens that ARDS has a mortality of 40%. But if the patient also has an acute kidney injury, AKI, then the mortality becomes from 40% to 80%. So AKI is a very severe condition when it is combined with ARDS. But most of the times ARDS management does not take into account of the AKI into that. So we have tried to apply metabolomics to understand how ARDS combined with AKI are different and why the mortality is so high. Is there any biomarker which we can detect in ARDS with AKI patient? And if we can understand the pathway, then clinician can tackle those pathways in the AKI condition. So this was one of the studies. So here, what do we have to do in terms of our study design? If we are going to study ARDS with AKI, so we have to take a two group. One group ARDS with AKI, one group ARDS without AKI, and compare the two. This is exactly what we did. And then the two group was ARDS without AKI, ARDS with AKI, acute kidney injury, and the number of patients was very less because it's very difficult to get patients and data. And then we found that this ARDS with AKI and without AKI can be easily differentiated by multivariate analysis. And these are the markers, biomarker of AKI comorbidity condition, for example, like creatinine, acetylcarnitine, arginine, histidine, methylhistidine, glutamine, glutamic, and cis acetonitrate. So In terms of mortality, survivor and non-survivor can also be looked into that. Mortality in ARDS with AKI. So these are the molecular marker in non-survivor group, ARDS with AKI, like isobutyric acid, valine, and cis-acetonitrite. Well, all these molecules are very good correlated with the clinical scores like Apache and so far. With that, and not only that, we can get a pathway study, like when the AKI is present with ARDS, then insulin metabolism, arginine, and these are the pathways which are mainly affected because of AKI condition. So clinicians have to design their study or the molecule to tackle these pathways, which when the AKI is present. So for the high mortality conditions, we have found that these are the main conditions which gets affected when ARDS with AKI is present, like it leads to acidosis, hyperammonia, hyperinflammation, high energy requirement, and mitochondrial dysfunction. And corresponding to all these conditions, for this condition, glutamate pathway is affected, which is related to urea cycle. And then for hyperinflammation, this beta oxidation is affected, and similarly for all the different conditions, TCA cycle is related to the mitochondrial dysfunction and the other part of that. So based on this some analysis, we can say that we have got a better understanding of ARDS with AKI through metabolomics. So take home message are two. One thing is, like, you have to design your study in such a way, and then it leads to biomarker. Biomarker is very good for getting an early diagnosis or better diagnosis. And then pathway analysis gives more understanding about the biological pathway. Now recently, metabolomics is also being used for drug discovery. You see, if there is a method by which you can understand the pathway, you can design some molecule to affect those pathways. That is what is one of the idea of using metabolomics for drug discovery. So it has been systematically studied, like you take a particular disease where you want to develop a drug, then do metabolomics, find the pathway, and then understand those proteins which are responsible for that pathway. And once you understand those proteins, you can design those molecules to target those pathways, and then do the metabolomics in presence of those molecule to understand whether the pathway is really affected or not. So this entire process has been streamlined, and people are rapidly using for getting a drug discovery. So instead of having everything blindly, the metabolomics with the drug discovery part can give more rapid and better drug discovery protocol into that. So there are different cancer metabolites which has been used for getting the better cancer therapeutic drug and targeted to that. So with this, I will be happy to answer questions, and feel free to ask any questions which you could not understand in my presentation.
Good morning, sir.
Good morning.
As per your slide, you saw about the serum. Sir, what is serum, and please explain about serum.
Serum is a component of blood. Okay. If you take a blood and allow it to coagulate, and then remove all the cells, the liquid which is left is serum. So, and serum is a very important biofluid because it has a lot of molecules apart from the blood cells, which gives the information about the patient from where it has been collected. Okay.
Thank you. Thank you. Thank you so much, sir, to enhance our knowledge about metabolism as a biomarker and give better understanding regarding the severity of the patient. Thank you, sir.
Thank you so much, sir. Now I request co-chairperson of 9th technical session of SLA, esteemed speaker to certificate and memento. Give a big round of applause.
Thank you.
तो आप सभी का बहुत बहुत धन्यवाद। अभी हमारे पास एक ओरल प्रेजेंटेशन है जो इसी सत्र में होना है। इनका एक्चुअली कल होना था, ये अनुपस्थित थी, इस वजह से इनका अभी कराया जाएगा। सो आई काइंडली रिक्वेस्ट टू आवर chairperson to kindly chair for the oral presentation of Miss Bhavani Sharma. So I request to Miss Bhavani Sharma to come on the stage and present their work. For your kind information, I will tell you some norms of the oral presentation. Your presentation should be 5 minutes and 2 minutes for the open discussion. So total presentation time is 7 minutes.
Good morning, all of you. Today, today's topic for my presentation is exploring the novel potentials of apoptosis and DNA intercalating activities of the immodine derivative. So before proceeding to the in details, first we will come to know the introduction part key what is immodine and actually it belongs to which class. It belongs to the anthraquinone means compounds belonging to the Polyphenaceae family, and chemically it is 9,10-dioxoanthracene core. These are the secondary metabolites of the plants, means they are helping the protection of the plants. And there are several antibiotics also. Anti-cancer antibiotics have been derived from the anthraquinone class of compounds like donorubicin, doxorubicin, apirubicin, which are already well established in the market with their potent anti-cancer activities. The structures are there. Now coming to the next slide about immodine. Immodine, actually, we are study part is my which have been extracted from the roots of the Rheum immodi. As in the presentation, you can see it has been extracted from the roots, which from the Rheum immodi, commonly known as the rhubarb. And these are the various sources and the pharmacological activities which have been already published in so many of literatures, which is already present over there. But for our research, we took the part Rheum immodi only. Now coming to the next slide, the mode of means what is the mechanism of action, how it shows the intercalation activity in the for showing anti-cancer activity. Actually, it goes through the caspase-3 mediated apoptosis in the cells, especially in the S phase of the cell cycle, and it belongs to the what is caspase? Caspase belongs to the cysteine protease family, and it projects its action on the aspartic acid part. And for this anti-cancer activity, we have taken the four this cancer cell lines. One is HEG2, which is the liver cancer cell line. PC3DU145, which is the prostate cancer cell line, and MCF7, which is the breast cancer cell lines. These are the majorly commonly used cancer cell lines, which are generally used in the studies. Now, coming to the next, how we did the extraction of the imodine. Imodine first, we took in the roots in the powdered form. Then we soaked it into a percolator for about 24 hours in ethanol. Then the percolate was collected, and this process was approximately done 4 times. And extract in the after doing the 4 times collection, it was around 200 grams. And this 200 grams, whatever extract of methanol we extracted, we done the chromatography against the silica gel, and silica gel the means the solvent used were hexane the chloroform in ratio 70 to 30. After doing this chromatography, we did the identification step that was done by the 10% H2SO4 in methanol extra solvent solution. And finally, we found that the orange spot is coming under the UV light, which is an identification confirmation that it is imodine only. How can we say that it is imodine? Because the literature is there that the orange spot identifies for the imodine. Coming now, proceeding to the next step. After extraction of the imodine, we planned for going for the synthesis of means chemical synthesis for its derivatives. So first derivation we went for the O alkylation, which was done in presence of the alkyl halides K2CO3 DMF at 60 degrees Celsius, and it was stirred for around 4 hours. Approximately yield it came 60 to 80%. Uh, uh, then these alkyl halides were alkylated products were derived. Then these are the in this slide you can see the different derivatives which we derived with the IC50 values against all the cancer cells which we took for our study. IC50 values, there are 5 derivatives. You can see it. Now, next, moving to the next slide is the esterification process. Second, this esterification. Why did we went for esterification? Because there are OH groups present over there. So we just got an idea. Okay, we just go for the ester derivation process. So for this ester preparation, we did cinnamic acid preparation derivatives like nicotinic acid preparation derivatives, and reagents and conditions used for the aromatic acids, DIDSM, DIPC, DIA at 0 degrees Celsius, and almost the yield were around 60 to 65%.
Please conclude your presentation.
Okay, sir. These are the LC50. Then third derivation means synthetic process was amide derivatives. Now coming to this final biological studies we did because we have to just get to know how much cytotoxicity we have to whether these drugs are safe or not, whatever derivatives we have done. So for these two studies were done apoptosis and caspase reactivation. Uh,
You just go on the your conclusion or summary slide.
Results.
Please.
Okay. Now the results and discussion from all these experimental studies, we got to know that our derivative 5 was the most potent based on the IC50 values, and it was compared with the apirubicin marketed drug which is already present in the market. So these are the values you can see derivative 5 and the apirubicin standard. So after studying all this one figures, we got to know that derivative 5 was almost comparatively potent to the standard drug.
Thank you. Now session is open for the discussion. Any question?
The co-alkylation and esterification will take place at same position. Alkylation and esterification will take place at same position in your slide. Takes place at same position.
No, sir. The position in the alkylation it was on the
Okay. How you can say this alkylation is done on that particular position where the hydroxyl group is attached, whether the imodate molecule to other hydroxyl group is attached. The carbonyl group makes chelation with hydroxyl group. How is this possible?
Because there is carbon group, it's so that we can see that Johnson 12 that was confirmed with the present. Then that
Means the actual activity is done by that hydroxyl group which is around the carbonyl group.
go for the derivative. Strategy that we can do go for that matter.
Or you can find the that substituted derivative of imodic is more active regarding your study.
As for the IC, in comparison to different market, compared both there were 10 derivatives, derivative 5 was the most.
What is the probable mode of action of that new derivative regarding this study?
We'll go for the thing.
What do you understand by apoptosis and what are the drugs being called apoptosis? Then do you name some drugs other than anti-cancer drugs? Is there any drug? And do you know about thalidomide? Thalidomide. Is it causing apoptosis or not? Okay, okay, thank you.
Now, this is the time for to thank the chair of this session. So let's thank our chairs.